Bismillah ar-Rahman ar-Rahim. Hello everybody. This is the lecture number 10, part 3, about the differential stain. We will explain about the gram or grams stain. Why called or named gram stain? That's why it's called as grams stain, because Danish bacteriologist Hans Christian Gram discovered this type of stain. It is very important stain in routine bacteriology. It divides by which the bacteria into two groups of bacteria, into gram-positive and gram-negative bacteria. The reagents that used in gram stain are solution 1 called primary stain, methyl or crystal violet or methylene blue. Why it's called the primary stain? Because the crystal violet or methylene blue is used as the first solution in a gram stain procedure. The solution number 2 is called mordant or fixative. It is logal iodine. Logal iodine. Why it's called mordant or fixative? Because it fixes the stain in the bacteria. The solution number 3 called Decolorizer. Decolorizer. Why it's called by this name, Decolorizer? Because it removes the color from the bacteria. The decolorizer or solution in gram negative called or is ethanol in a concentration 95%. The solution 4 called counter stain or secondary stain. The red or the color of the counter stain is red, so we can use neutral red or safranine or carbolfoxin. So in a gram stain we use 4 solution or 4 reagents in 4 steps to stain the bacteria. Solution 1, primary stain, which is the crystal violet or methylene blue. Solution 2 called mordant or fixative, which is logal iodine or logal's iodine. Solution 3 called decolorizer, which is ethanol 95%. Solution 4 called counter stain or secondary stain, which is neutral red or safranine or carbolfoxine. Now, list the steps of a gram stain. Or describe the steps to make the gram stain. Other question: when should you use gram stain and why? And why? The procedure of a gram stain is: prepare the bacterial smear, allow to dry and fix with gentle heat. You can take or make the bacterial smear either from the bacteria which grow on petri dish, or you can take a drop of contaminated water or water to know what the species of bacteria or genus which are present in the water, or you can take the sample from milk, from swab, to make the bacterial smear by using lube. After fixation, by passing the slide 3 times on the Bunsen burner, now the bacterial smear become ready for staining by gram stain. Firstly, put crystal violet or methylene blue on the bacterial smear and allow it for 1 minute. After that, wash the slide with tap water or distilled water. Add the solution number 2, which is Local's iodine. The color of Local's iodine is brown. Let the local iodine for 1 minute on the bacterial smear. After that, rinse the slide with tap water. Put the decolorizer on the bacterial smear for 20 seconds to 30 seconds and wash the slide quickly. The last step, add the basic foxin or carbolfoxin for 1 minute and then wash the slide with tap water or distilled water. Then examine the slide under the light microscope by using oil immersion. Gram-positive bacteria appear blue to violet, but gram-negative bacteria appear red in color. This unstained bacteria, gram-negative and gram-positive bacteria. When you put crystal violet, the gram-negative bacteria and the gram-positive bacteria will appear blue. When you add the decolorizer, the color removed from gram-negative. Finally, gram